Hello everyone, this is the paper presentation for General Club. The title includes the site mapping and small molecules blind docking that reveals a possible target site on SARS-CoV-2 MPRO dimer interface. Introduction. On December 31st, 2019, World Health Organization was informed of cases of pneumonia originating in China. Outbreak was quickly identified to be caused by novel coronavirus, which is causing COVID-19, resulting in an ongoing pandemic with serious health, social, and economic implications. On the basis of genetic relationships, including SARS-CoV-1 and MERS-CoV, introduced in humans in 2002 and 12 respectively, novel coronavirus has been designated as SARS-CoV-2. Aim of the study was to investigate the small molecules including ritonavir, alpha-ketonemide and epsilon for their ability to interact with MPRO of SARS-CoV-2 and to explore the interactions of small molecules with dimerization pocket at apex of MPRO. Materials and methods. The step first include the docking to the active site of the virus MPRO. Docking calculations were performed using QPLD workflow, which aims to improve the partial charges on ligand atoms. Protein structures were prepared using the protein preparation wizard, and then ligand structures were processed using ligprep to obtain the structures with optimized geometry. Step 2 includes the mapping potential ligand binding sites on the virus MPRO using Stongier sitemap tool and Prank Web server. Figure shows the docking of ritonavir, alpha-ketonemide and SRT, the three compounds that were under the study to active site of the virus MPRO. Compound structures are shown in figure A. Figure B depicts the compounds that are docked to the active site of monomer using the glide, and C shows the blind docking performed using autodec. Figure D shows the docking to active site of the dimer using the glide, which is the empirical scoring function to approximate the ligand binding free energy. This figure shows the sitemap analysis of main protease in its monomeric and dimeric form with the catalytic residues, that is histidine and cysteine, at position 41 and 145 of active site respectively. Hydrogen acceptor and donor are indicated in red and blue colors, while hydrophilic and hydrophobic regions are in green and yellow colors. Step 3 includes the blind docking using Autodeck. Protein and ligand structures were processed to generate PDPQT format, which is very similar to PDB format, and includes the partial charge storage and atoms of Autodeck 4 type. Step 4 includes the MD simulations using GROMEX software with CHARM M27 force field. Energy minimizations was performed using the steepest descent gradient method to iterate for finding a optimized local minima of differential functions. Topology of the docked ligands was generated using SwissPalm, which provides the parameters for the three organic molecules that are under the study. Then the bond lengths were constrained using Linux algorithm, and free energy calculations were quantified using GMMPBSA tool. This figure shows the blind docking of the three compounds in A, B and C part to the MPRO dimer. Binding affinities are stated along with the interactions diagrams that depicts the hydrogen bond formation. Proximity of ligand to active site is indicated by the catalytic diode residues, that is histidine and cysteine, at position 41 and 145 respectively, while proximity to domain 3 dimerization apex is indicated by residue alanine at 285 position. Results and discussion. Docking to SARS-CoV-2 main protease active site highlights the high throughput binding affinity for structurally diverse small molecules. Study findings indicated that the three molecules that were under the study bound to the active site with glide scores with respective values, and the compounds formed hydrogen bonds with key residues of protease active site at the three different residues at three different positions. The three compounds were shown to have the preference for binding to active site. Top ranking POSes produces the respective binding affinities, with 10th, 18th and 17th POSes find to bind in the active site in the monomer. This figure shows the comparison between the structures of virus M-Pro with three different PDB IDs. A shows the protein structures that are aligned with qualitative similarity scores, and B shows the root mean square deviation between the structures that are separated on per residue basis. C shows the binding sites for main protease that were compared between the structures in each protomer for PDB ID 6Y2G, and these RMSD values shown are for carbon alpha atoms of residues. The figure shows the average contributions that were decomposed into a per residue basis for binding of the virus M-Pro with the three different compounds to the active site in protomer A and active site in protomer B and domain 3 apex. This figure shows the comparison between the docking to active site of the virus M-Pro structures with three different PDB IDs in both protomer A and B. Hydrogen bonds are indicated by yellow color, pi-pi stacking in cyan, and salt-rich formation by magenta lines. MD formulations highlight the stability of ligand complexes in active site and in domain 3 of SARS-CoV-2 M-Pro. Protease complexed with ligands in active site shows the respective RMSD values for three compounds, that is ritonavir, ketonamide, and SRT 1720 for both active site and domain 3. And these values suggest that binding to active site may have a modest stabilizing effect than domain 3. This figure shows the stability of main protease complexed with ligands bound to the active site and domain 3. Values for apoprotein are shown in blue color, ritonavir, alpha ketonamide, and SRT in purple, red, and green color. Part 1 shows the average root mean square deviation values that were calculated for the protein with respect to backbone, and part 2 shows the average RMSF for whole protein that was calculated, and part 3 shows the RMS difference that was calculated by subtracting the values of apoform from ligand bound form of the protein. Conclusion: the complexes were analyzed using MMPBSA to calculate binding free energy. The three compounds that were under the study, out of these three compounds, alpha ketonamide and SRT bound strongly with the free energy to active site, and ritonavir bound strongly with the binding energy with domain 3 then to the active site. Overall site mapping highlights a highly reactive binding pocket at domain 3 of the virus M-Pro, and this suggests that the binding to active site may have the stabilizing effect than domain 3, means the attraction of the small molecules to the site is consistent with the blind docking analysis. These are the references. Thank you so much.